So for Chapter 3, Segment 4, it starts with the section Receptor Molecules Recognize Transmitters. The first item that is discussed is cholinergic synapses, which refer to the cells that use acetylcholine as their synaptic transmitter. There's two different kinds of these, muscarinic or nicotinic cholinergic receptors. Muscarinic cholinergic receptors are mostly in the brain, and they're also found on organs that are innervated by the parasympathetic division of the autonomic nervous system. They contain both excitatory and inhibitory causes. And then for the nicotinic cholinergic receptors, they have synapses at muscles in the autonomic ganglia. Most of these are excitatory. The anatomy of the nicotinic acetylcholine receptor is important. It's a lopsided dumbbell with a tube running down its central axis. The handle of the dumbbell then spans the cell membrane, and the sides of the ion channel then run through this handle of the receptor and consist of 5 protein subunits. 2 of these subunits are then alike and provide 2 recognition sites for acetylcholine. In order for this receptor to be open, both sites must be occupied. So one of the experiments that was done in order to prove that the nervous system uses both chemical and electrical signals was done by Otto Louis. The experiment was he stimulated the vagus nerve of a frog, knowing that this would decrease the heart rate, then collected the fluid that was around the heart, then bathed another frog heart in this fluid, and then the other frog's heart rate slowed like the first frog, and the result, he believed, was that a chemical was leaked into this fluid, being later proven to be acetylcholine. The coordination of the different transmitter systems of the brain is extremely complex because each subtype has its own unique pattern of distribution. Different systems are turned on in different points of the time or day or in the life, and there's a number of some receptors that vary with use with drugs. It could be either upregulation or downregulation for the coordination of the different systems. Upregulation being a compensatory increase of the receptors of the synapses of neuron, and downregulation being a decrease of the receptors. Next section is transmitters bind to receptors and they gate ion channels. The recognition of transmitter molecules by receptor molecules controls the opening of ion channels in 2 different ways, which are ionotropic receptors and metabotropic receptors. Ionotropic receptors are ion channels that are opened by being bound by an agonist. These directly control ion channels, and they're commonly referred to as ligand-gated ion channels. The other type is metabotropic receptors, which don't contain an ion channel, but may, when they're activated, use the G protein system to alter the function of the postsynaptic cell. So overall, they recognize the synaptic transmitter, but don't directly control the ion channels. They do this through G proteins for the most part, which are a class of proteins that reside in the intracellular portion of the receptor that are activated when the receptor binds an appropriate ligand on the extracellular surface. Sometimes this G protein acts itself to open the ion proton, or it affects another internal signal to open an ion channel. If you consider the first messenger to be the neurotransmitter, the second messenger would then be this electrical signal from the G protein, and the benefits of it are that it amplifies and prolongs the synaptic signals in order to open up the ion channels. The process with the G proteins are that the transmitter then binds to a receptor, the metabotropic receptor with the G protein, and then part of the G protein complex separates from each other. The alpha subunit in particular migrates away from the cell and modulates the activity of the target molecule. The next section involves the action of synaptic transmitters is stopped rapidly. Particularly chemical neurotransmitter postsynaptic effects are prompt and brief, which ensures the message to be repeated. In order for this to be achieved, most of the time, accurate timing of synaptic transmission is necessary. This brevity of the transmitter effects is achieved in two ways, either by degradation, which is the breaking down of the chemical neurotransmitters into inactive metabolites, or the reuptake of the neurotransmitter in order to be repackaged into a synaptic vesicle to be reused to excite the postsynaptic membrane once more. Next section is in regards to several factors that regulate neurotransmitter release. The first one discusses autoreceptors, which are receptors in the synaptic transmitter that are located in the presynaptic membrane. They tell the axon terminal how much transmitter has been released. So it's basically auto-recepting itself, so it's targeting itself to give a signal. And this can control the amount of neurotransmitter that's released and control the concentrations within the synaptic cleft. Two of the simplest synapses are the axodendritic synapse and the axosomatic synapse, where the first one is an axon synapsing directly onto a dendrite of the postsynaptic neuron, and the axosomatic synapse is the axon synapsing onto the cell body of the postsynaptic neuron. There's also different types that are less common, but the axoaxonic is axon onto axon of the presynaptic to the postsynaptic. This allows the presynaptic neuron to regulate how much neurotransmitter will be released from the targeted terminal. Another one is a retrograde synapse, which a signal flows from the postsynaptic to the presynaptic, so it's counter to the usual direction of synaptic communication. Because of this, transmission starts with axodendritic synaptic activity, then the postsynaptic cell releases a gas neurotransmitter, which this signals the presynaptic cell to release more transmitter. So it's kind of like a regulatory system to release different levels of transmitter. There's also dendrodendritic synapses, which is a connection forms between dendrites of two different neurons, which helps with the coordination of activity. There is ectopic transmission, which is cell-to-cell communication based on the release of neurotransmitter in regions outside of traditional synapses. These are formed between many neurons and the location of the transmitter release and sites at which the transmitter acts outside of conventional boundaries of nearby synapses. The last one that we're discussing is varicosities, which are axonal swelling from neurotransmitter diffuses into a non-directed synapse. Non-directed synapses then are a type of synapse in which the presynaptic and postsynaptic cells are not in close apposition. Instead, neurotransmitter is released by axonal varicosities and diffuses away to affect wide regions of tissue. It allows for steady release of neurotransmitter to broadly affected areas. Last section that we're discussing is neurons and synapses combine to make circuits. Circuits then are a group of neurons and their synaptic interconnections. Neurons have two different types of processes. They can either have analog-like signals that vary in strength or digital-like all or none signals that vary in frequency. A neural chain then is a simple kind of neural circuit in which neurons are attached linearly end to end. The knee-jerk reflex is an example of a neural chain which goes from the sensory neuron to a motor neuron with only one synapse for communication. The reason that they're so fast is that because this reflex is myelinated, has large diameter nerves, and it has sensory cells that synapse directly onto the motor neuron. Last two are revealed to us through the neural chain within the visual system, which includes convergence, which is the phenomenon of neural connections in which many cells send signals to a single cell, and divergence, which is the phenomenon of neural connections in which one cell sends signals to many other cells. So with the eye, there's like one nerve and it splits out into many different neural connections, which is convergence, and then, or sorry, divergence, and then convergence, it all goes to one point. And that is it for chapter 3, segment